Okay. Good morning. All right, let's get started. All right. Okay, I had no idea the white screens had to get up to make this work. Learning things, very cool. Okay, hope everybody's doing good. And you have me 2288. You only have me 2 more lectures, 2280. You have me, I think, 5 more lectures. I hope you'll survive. But we're more than halfway through, so we're almost there. And I hope you guys can follow along. I'm trying to keep up with the forum, so I usually answer questions in the morning, sometimes the day after, depending on how fast I get to it. But in general, I'll try to keep up to speed with it. The forum has good questions posted already, and I always try to mention things in the lectures again as well to clarify any questions that are there. As always, you're welcome to ask questions anytime. Okay, you can interrupt me. I really don't mind. You can also remind me to slow down. I know I tend to speed up, and I'm grateful if people remind me. I really don't have any issues with that at all. I think this is kind of where we stopped last time. Does that seem right? Okay. All right. Thanks. Okay. So just to give you a second to get back into it, we were talking about translation and how proteins are translated from an mRNA into a protein sequence, or how sequences are translated. And we talked about how eukaryotes recognize the translation start site as proximity to the cap protein. And bacteria have a little bit of a different way of recognizing start sites. They have a specific sequence they recognize, similar to a promoter sequence, only that now we are talking about an RNA sequence here that's upstream of any gene that they want to translate. So this explains, and I think that's what I left off, how bacteria can translate several sequences within one RNA. Eukaryotes cannot do that, okay? Eukaryotes always have to start from the cap binding protein to the first AUG. So they can only do one per sequence. Whereas bacteria, because they recognize a specific RNA site, that it can be anywhere in the RNA, there could technically be 20 proteins within one RNA. Only that the RNA would then get very long, so they usually keep it shorter, but there are longer operons than 3. Which, we had this one, brings us to the function of the ribosome. So the ribosome is actually pretty cool in terms that, you remember that we had a big structure made out of RNA and protein. So the catalytic sites, or the things that are close to where action happens, are all RNA. So it's not actually the protein. We always think of enzymes or things that do the work being the proteins. In the case of the spliceosome and in the case of the ribosome, it's actually the RNA structure that does all the work. And that's in both bacteria and eukaryotes. So that's one of the things that researchers who like the idea of having an RNA world bring as an argument for the presumed existence of an RNA world. So an RNA world would be a world that existed prior to our current, what we call protein world, where most activities or most enzymatic activities are carried out by proteins. They think back in the day, there was an RNA world where we just had RNA doing all the jobs. I'm against this theory, but it's like anything that you have no proof for, like is there a God or not? You can choose this yourself, whether you believe in an RNA world or not. That's up to you. And you can feel free to join the online community of 4. It's kind of like people get very heated about that. So there's some profs in the biochemistry department who are all for RNA world, and they're always anti-RNA world. So we have nice discussions about that. Semi-nice, kind of, depending on how many beers we had. All right. So that's your ribosomes. they recognize the in bacteria, they recognize the Shine-Delgado sequence, which is the sequence in the mRNA that base pairs with the rRNA. And then that's what allows the ribosome to bind to the sequence right upstream of the AUG site. Okay, which finally brings us to something that I haven't really mentioned yet, which is elongation, how we've had initiation, how things get started. Now we get in how do things get carried on. And I mentioned that most of the energy that we need to form this polypeptide bond comes from the amino acid tRNA that we have in the beginning. Well, there's a little bit more energy used. It's not directly for the formation of the peptide bond, but we have so-called elongation factors, or EFTU, that bring in the new amino acid tRNAs to the ribosome. So while we like to think that the tRNAs, the charged tRNAs, just kind of float around, they don't actually just float around in the cytoplasm because they're so high energy. If you remember, the ATP activates the amino acid, which then gets charged on the tRNA, and this is a really high energy bond. You can't let things float around that are that high energy, so they bound to so-called elongation factors, which brings them to the ribosome. So that's all I need you to remember. I don't need you to remember exactly what happens here. I just want you to remember there's more factors that help in charging the ribosome with the amino acids. Okay, and then finally, termination. You remember there are 3 stop codons in that genetic code table that we had. So termination doesn't just mean it doesn't code for no amino acid. So it doesn't just mean that, okay, we run, let's say the ribosome runs into a stop codon. It doesn't just stop on its own. It needs help to release itself from the mRNA. So if it runs into a codon that it doesn't find the tRNA for, the ribosome will just sit there. It won't go anywhere. Okay, so it can't release itself without any help from the mRNA. It needs so-called release factors. So there are no tRNAs for stop codons, right? Because we don't have an amino acid that is responding to this codon. So we need specific release factors that will bind to the stop codons. There's 2 in humans, there's 2 different release factors in bacteria as well. And they recognize the different stop codons. Okay, so basically they are, you can see this is the structure of a release factor. And to the cell, this looks like a charged tRNA. And what you see down here is what the cell or the ribosome thinks of as the anticodon. So it finds something. I know it doesn't look like a charged tRNA for us, but if you're very imaginative, you can maybe, it fits right into the ribosome where the tRNA is supposed to fit. And that allows the ribosome to dislodge or dislocate from the mRNA. Okay, so what happens is the ribosome runs into this mRNA stop codon, and then the A site remains empty, right? It can sample different tRNAs, but it remains empty. It can't continue its function. And instead, a release factor then can bind into the site. And this is after enough sampling, the ribosome sits there long enough, eventually a release factor will get sampled. And the release factor allows the peptide chain to be released from the tRNA. And then the tRNA gets moved out of the ribosome, and the release factor has to be released from the ribosome as well. So what I need you to remember is that you need a release factor in order to stop translation. It doesn't just happen on its own. And this step also requires some energy. So we need to hydrolyze GTP into GDP in order to get everything off together. So why do we need a release factor? Why can't we just, if we don't have a tRNA that matches, why can't we just go off on our own? The problem is sometimes the ribosome doesn't find the right tRNA right away. Let's say it's trying to find a tryptophan tRNA, like that has a tryptophan charge to it, but it's just not around, or it's just rare. We can't find it right away. That would mean anytime the ribosome sits too long, it would release itself from translation. We don't want that. We want translation to really only stop at a stop codon, so that we don't end up with all this prematurely stopped polypeptide. So we need the release factors in order to terminate translation. Good. Yeah, question? It needs to be transferred to something to be chemically released from the tRNA. Remember, the end of the peptide is chemically attached to the tRNA, so you need to have a chemical reaction to get it loose. And what is a simple one? Yeah. Good. Okay. So how accurate do you guys think translation is? You think it's good? Does it have to be good? Does it have to be as good as replication? You can see the numbers here. It's not as good as replication. So you can see replication has an error rate in humans of 10 to the minus 8. Okay, that's the chance of making a mistake in replication. And I just heard a talk where somebody said that between two people, everybody has about 50 new mutations that they didn't inherit from either one of their parents, and those would be replication errors. Okay, so they knew they not just between you guys different, but between you and your parents. So we all have new mutations which make us unique and which allow for evolution to continue. Okay, so we can continue the path of becoming more. I don't even know what we're going to become, but otherwise we would still be on trees. Okay, we need mutations in order to get evolution to go. Transcription doesn't have any impact on offspring, so there can be a higher error rate allowed, so it's 10 to the minus 5th. And then because if you'd make one mistake in one mRNA, then that mRNA would be translated differently, but it doesn't have any implication on the future. And most likely, most of your other mRNAs don't have that same error. So you can have some mistakes allowed in transcription. Now, translation, even worse, you have an error rate of 10 to the minus 4. Okay, so you have more errors in translation than you have in replication, because it's okay. If we make let's say we make 1,000 proteins from one mRNA, if there's 1 or 2 mistakes in there, or it's even less, it's only 10 to the minus 4th, that's okay. The proteins can still be functional, or at least 999 would be functional. If one is not functional, the cell can live with that. So how does translation happen, or how do those errors happen? Those errors are called mistranslation. And they can happen by either wrongful base pairing here or by wrongful amino acylation. If we have the wrong amino acid attached to a tRNA, that will automatically get included in the peptide. Or if you have a mismatch in base pairing, that will also get included in the peptide. So those are your main means of mistranslation. So mistakes are being made. And most often they are being made at the charging of the tRNA. So here's your amino acid tRNA synthetase. It has to pick the correct amino acid from all the amino acids in your cell. And as I said before, it's not just the 20 amino acids that you know. Those are the ones that go into proteins. There's many more amino acids that are in the cytoplasm that are not proteinogenic amino acids. So it has to pick the correct amino acid and has to pick the correct tRNA in order to get the correct RNA charged. You can see here the means of making a mistake, or the ways of making a mistake, is picking the wrong amino acid in this one. I picked yellow instead of green. And here it picks the wrong tRNA. So you could have the wrong tRNA bound, even though you have the correct amino acid. If you put it on the wrong tRNA, that will also lead to mistakes further down. Many synthetases have a proofreading function where they double check whether it's correctly ligated, and they will kick out the ones or cleave the ones that are not correctly ligated, but not all of them do. So mistranslation happens all the time, but usually it's not detrimental. Okay, usually the cell can live with some amount of errors. So you don't need to memorize this. I want you to remember that you can pick the wrong tRNA or the wrong amino acid. Okay, so you have those 2 chances of getting the wrong thing from your whole pool. And that just shows you how good those synthetases have to be in order to be accurate. Which brings us to inhibitors of protein synthesis. So some, as I mentioned before, there are differences between eukaryotes and bacteria. And eukaryotes and, us, bacteria, can be pathogenic. Anything that's different between bacteria and eukaryotes can be used as a point where we try to use antibiotics to just kill bacteria and not the eukaryotes. So we don't want to damage our own body while we fight off the bacteria. We don't want to add insult to injury, basically. Again, please don't, as I said, I will not, I will tell you when you don't need to memorize things. You don't need to memorize this table. But I want you to have a good look at it so that you get an idea of how different things can be. So you can have inhibitors of translation initiation here. There's 3. Streptomycin is one that you guys may have heard about. It prevents the formation of the initiation complex in just bacteria. So that's why it says prokaryotic here. You can also have tetracycline and again streptomycin and chiromycin, for example, are the ones that again inhibit elongation in just bacteria. And then elongation, this one we use a lot, chloramphenicol, we use that a lot in the lab, inhibits bacteria in the elongation. You can see it binds to the 50th subunit, blocks the A site, and inhibits peptidyl transferase activity. Okay, so you can see now that you've learned all this, when you read a table like that, these things should actually mean something to you. So all of what you learn here has some meaning, even to people who don't study science, because they take antibiotics or there's something that affects them in their life. Now, some that we don't like, which is not an antibiotic, is diphtheria toxin. So diphtheria is one that inhibits, it's a bacterium that excretes a toxin, and it inhibits the eukaryotic one and not the bacterial one. And that's why we get diphtheria. Okay, so it works the other way around too, that bacteria come up with something that damages the eukaryotic cells. And this, interesting enough, this is actually a little protein. So it's not a small chemical like streptomycin and chloramphenicol, those are small chemicals. Diphtheria toxin is a protein that inactivates EFT elongation factor 2, which then inhibits translation in eukaryotes. And then you have ricin, which is a well-known poison and has been sent out in letters. It's very, very toxic. And again inhibits eukaryotic translation by inactivating the 28 srRNA. So you can have different, poisons for us would work the same. So if we can't do any translation, that also means we can't build any new cells, and usually that leads to quick organ failure. Usually works on the mitochondria first, so you don't have any more energy in your cells die, you die. So it goes both ways. So remember that any difference in eukaryotes and bacteria can be exploited, but by either, by either side. Okay, good so far for translation. Yes, question? Which one? This one?
The next, this one, yeah. So I think you mentioned how mistranslations are 2 very common ones. So you might just read that passage.
Okay, so the 2 very common, okay, so you can have mistranslation happening by having the wrong amino acid charged to a tRNA. And that can be either that the wrong tRNA is recognized or the wrong amino acid is recognized. So that's your first possibility. The other one would be that the base pairing here doesn't work properly. And then if the base pairing doesn't work, you will also get mistranslation. So problems with base pairing are usually if the wobble doesn't work correctly, then you have issues with translation down the road. Okay, so those are your the two, when we talked about translation last lecture, I think, I said the 2 means of accuracy is the charging of the tRNA with the correct amino acid, and then the anticodon-codon binding. Either one of those don't work properly, you get mistranslation. Okay, good. With translation so far? Because we're done with translation after that. All right. Okay, so now what happens with the proteins after they mate? Many proteins after they mate are ready to do their job. Okay, so not all of them need to have some special treatment, but some of them do. But most of them have to fold into a specific structure in order to be active. Okay, so life and death of proteins. Proteins need to be folded. All proteins need to be folded, but most of them don't need help. They can do it on their own, okay, just by the primary amino acid sequence determines their secondary and tertiary structure. Many proteins need to be modified. That means they need to be changed chemically after they're being made. And in the end, all proteins need to be degraded. They need to be gotten rid of eventually. So why do proteins need to be degraded? Because they do sometimes change over time just by chemical insult, by having done their job many times and having little chemical modifications along the way. And sometimes we just don't want them anymore. Okay, we need them for a specific purpose, but then we don't want them anymore, so we want to go degrade them. So some is active degradation because we don't want their function, and some is degradation because they damaged and we don't want them anymore. Okay, so protein folding. We discriminate between primary structure, secondary, tertiary, and quaternary structure. It's pretty simple. So your primary structure is just the sequence of amino acids that you have in your protein. That's, determined by the mRNA sequence, which is determined by the DNA sequence. Am I cutting out? It sounds like I'm cutting out. It's good. Okay, let me know if it's cutting out. Sometimes the battery dies. Okay, secondary structure. The secondary structure is actually largely determined by the primary structure. So depending on the amino acid sequence, you get alpha helical structures or beta sheets, or you get coils, which are unfolded sequences in between that can connect parts. So the secondary structure, we can predict that. There's different prediction programs which take the amino acid structure or amino acid sequence and tell you what this protein will most likely fold into. Okay, so it can be a certain amino acid sequence will always fold an alpha helical structure. So secondary structure also determined by primary structure. Tertiary structure is then determined by the secondary structure, largely. Okay, so it's basically you have your helical structures here, meaning you have in a helix, you always have the amino acid side chain sticking out to the side here, and that determines how they interact with other portions. That doesn't mean that we can predict a tertiary structure. We cannot. It's too complex. We can try to predict it, but most of the programs are not very good. So if you can predict it, and then you do a crystal structure and it looks totally different. The secondary structure we're usually pretty good about. Those programs are good. Tertiary structure, not so good. Quaternary structure is then how different proteins work together, and they can be either, in this case, it's almost it's a quaternary structure, which can be of four of the exact same protein, or they can be of different proteins. So a quaternary structure would be the ribosomal structure. Different proteins coming together with RNAs, that's your quaternary structure. Okay, so how can these proteins fold? There's three main pathways. The first one is the protein folds on its own. So this is supposed to be the ribosome. It comes out of the ribosome and folds itself. Okay, no help needed. Some proteins need a little bit of help. So there's HSP70, heat shock protein 70 is one of the proteins that help a little bit. They're chaperones. They will go through that in a little bit, but they bind the amino acids when they come out. And then afterwards, they slowly release them so that they can form their secondary and tertiary structure. And then there's the ones that's 85% of your proteins, and this one's about 70%. So most of them just do it by themselves. Okay. And then there's the ones that really need help, which are bound first to proteins when they come out of the ribosome. And then they go into this tub called GROYEL and GROYES. And then they come out of this tub perfectly folded. Okay, so they need to go into a folding machinery in order to get the correct structure. Okay, this is your GROYEL chaperoning complex. The majority of the proteins that go in here have some large hydrophobic regions. So if you have the protein coming out of your ribosome, the ribosome is just open in the cytosol, so it's hydrophilic, right? It's a watery environment. If the protein has large regions that are hydrophobic, they don't like water, they won't fold correctly in an environment that has water in it. So this big tub complex, the GROYEL-GROYES complex, creates an area where there's a reducing and hydrophobic tub that the proteins can form correctly in. So it's usually really a biophysical problem why the proteins don't fold correctly. Okay, and it's the same with the ones here. These ones, if they have some hydrophobic sections, they need to wait for the translation of another hydrophobic section in order to fold back properly. So it's a biophysical thing. They can't do their folding without having the whole protein. And these ones can't do their folding at all because they need to go in an environment that allows them to fold properly. Okay, this is your chaperoning reaction cycle. And what I want you to take away from this is that it needs a lot of energy to fold proteins that need to go into the chaperoning complex. So it needs 7 ATPs in the end. So you have the chaperoning complex has 2 tubs that are back to back. They will take one unfolded protein with an ATP, and then they will close the tub with GRO-ES, that's the top section. And then they hydrolyze their ATPs and fold the protein inside. So this is a very quick reaction, but it costs a lot of energy. So protein folding is not a cheap reaction for the cell. And then it sticks. The reason why this is drawn as 2 sections, it sticks in here, and only when the second one comes in does the first one get released. So you need a second protein to come in, go into the second tub, and then the first one can get released. Okay, and then it goes back into the cycle. So basically, you need to have one of them is working on making a protein, and only when the second one then starts up can the first one release its protein. So it's a dual tub complex thing. Okay. All right. So remember, it needs it has the tub, it has the lid. That's GRO-ES. It needs a lot of ATP in order to fold itself. And it's a whole complex cycle of having 2 tubs that work together in order to keep the folding going. That's your take home from this. Okay. Okay, so you don't need to draw this. So you do need to know that it requires ATP and 2 different sections to fold the protein. Okay. Chaperones are the proteins that help cells fold their proteins. So there's the chaperoning that I just showed you. But there's also just proteins that bind to unfolded proteins when there's danger. Okay, so sometimes, okay, your E. coli, the one that we work with in the lab, really likes growing at 37 because it was isolated from the gut where it's always 37 or about 37. So we grow them at 37, they're totally happy. If we heat shock them to 42, which is like a really good fever, the cells get very unhappy and they barely survive. They don't survive 45, but they're unlikely to ever encounter 45 because then you're dead. So, which, you know, then they're dead in the long run as well. So at 42, some of the proteins that they use on a day-to-day usage start unfolding. They're not stable at those high temperatures because that's not their optimal growth condition. So if the cell encounters a so-called heat shock, it starts expressing heat shock proteins. And heat shock proteins are mostly chaperones because they are protein folding helpers. So they can bind to proteins even after they're made. So it's not just binding them when they come out of the ribosome. It's also encountering unfolded proteins in the cytoplasm. They can bind to them and help them fold back into their optimal condition. So that's why all the chaperones are called HSPs, heat shock proteins, because that's where they were first discovered. So if you were to look at the proteome of a E. coli at 37 and one at 42, all of a sudden you have all these new proteins appear that are just there to save the bacteria from totally breaking down. Okay, so 2 prominent proteins that are not heat shock proteins, that are the ones that help with the ones coming out of the ribosome, are called trigger factor and nascent chain associated complex in eukaryotes. I don't need you to memorize the name, but they come up in the book all the time, or in the books and in the supplementary material. So these are chaperones that help the cell with their regular day-to-day making of proteins, things coming out of the ribosome. They bind there and help fold the proteins correctly. Does that make sense? That you have so you have 2 different times that you need chaperones. One is when proteins are being made and they can't fold on their own, then you need chaperones, or when the cell encounters a heat shock then lots of proteins may not be able to keep their tertiary or quaternary structure, and then chaperones come in and rescue them. Okay, which brings us to one of the most interesting proteins I find when it comes to protein folding. Anybody heard of prions? What do you say? Prions or prions? Prions? Okay. So I'm German, as you may have noticed with my accent that I always get comments about. We say prions. So if I say it wrong, it's because you know, takes me time to learn. I'll never learn it. Never mind. Okay. So prions are the infectious agent in mad cow disease. And they also cause transmissible spongiform encephalitis. That's also in a form of Creutzfeldt-Jakobs in humans. That's transmissible from cows to humans. I don't know if you guys are old enough to remember the huge burning of the cows in England. I don't even quite remember which year that was, but they had all these cows that went crazy, and they had to burn them in order to prevent transmission to humans. So that's prion disease. Prions are really cool because it's not a virus, okay? We know, everybody knows viruses now. We know bacteria, they're different from viruses, but this is a disease caused by a protein. So it's not, you know, viral transmission or anything like that. It's a protein disease. And what happens is there is a protein, and this we have in humans as well. It's called major prion protein, PRP. And you can see it's here in green. That's the regular form, and we find that in the brain a lot. So we don't know its actual function very well, but we do know that it's expressed in the brain and in the nervous system, but in the brain as well. Okay, so when this protein encounters a misfolded version of itself, the PRPSC, it changes its conformation to the one that the misfolded one has. And then this one contagious one makes a non-contagious one contagious. Okay, so that's how it goes through the body. And then in the end, you end up with just the misfolded ones, because any misfolded one can turn the folded one to the dark side. It's kind of like the Jedi's. Okay, so these are your two structures. So this is your normal version, and this is your diseased version. And it's the same amino acid sequence. So remember when I said you can see or you can calculate the secondary structure from the primary structure? You can, but it's not always, you know, if I would put this into the calculated structure, would probably come up with this one. But a small change in folding can make it into a completely different function. And then this is the one that it turns into. So these ones have a normal function, but when they encounter a misfolded one, then they also get misfolded themselves. That's how the infection precipitates. Yep. So yeah, so question, is it known how one induces the other to misfold? It's like any other protein-protein interaction that can cause a conformational change. It goes like that. So I think both forms are energetically favorable, but it requires a little bit of energy or interaction to go from one to the other. So the normal one will go to the other one when it interacts with that. It's kind of how you have what do we have? The cap protein interacts with the RNA polymerase, and the binding of it makes it more efficient titrin binding. So it's just the protein-protein interaction that changes the conformation. No, yeah, the normal one doesn't turn it back. That would be nice, right? Okay, so what happens with PRPSC is it's not as stable. It starts aggregating and accumulating, and you get those deposits in the brain. So if they cut up the cow's brain or the Creutzfeldt-Jakob disease person's brain, they can see abnormalities in the brain structure. And you know that's why the cows begin acting weird, because the brain is being damaged. So it's basically a form of brain damage. Okay, questions for protein folding? No? Okay, so remember when we say we can, predict structures, we're not always 100% right. So be careful with anything bioinformatics gives you. Usually you have to prove it in the lab later. And even then, we would have never thought one could move to another. Okay. All right, if we're good with protein folding, we'll go to protein processing. Protein folding is also part of protein processing, but the ones that we're talking now, or that we'll talk about now, are post-translational modifications. So post-translational modifications aren't just folding, so folding can easily be undone, right? It's something that can be unfolded again if needed. Protein processing are covalent changes like phosphorylation, acetylation, how we had it for the histones. They're real changes made to the primary structure of the protein. So your amino acids are going to be modified and changed. That's what a post-translational modification is. There's many different post-translational modifications. Some are the addition of sugars, which is glycosylation. Phosphorylation is the addition of phosphates. And those are some of the most prominent examples, but there's many different other ones. Okay, so you can add small proteins, say like ubiquitin to your protein. You can add whole lipids to your protein. There's many different things you can chemically add to a protein, and any chemical change will fall under post-translational modification. Proteolytic cleavage is the most common form because in the end it happens to every protein. Okay, in the end, even if you've never added a phosphate to your protein, all of them will be degraded. So that's why it's the most common one, because it will always happen to the protein. And so it's again the same. Why is it a modification? Because can't be taken back. Once you cleave it, once you cut it up, it's cut up. So anything that's a permanent change to your protein is a modification. Okay, so the R groups, you learned this with Dr. Bofa, that all the amino acids have the R groups, the site groups, so not part of the peptide backbone, but these are the ones that can be modified. So the R groups of your amino acids are the ones that can be modified. And you can see here, glycosylation is the addition of a sugar. So you don't need to draw the structure, but you should know glycosylation, the word glucose is in there. That means addition of a sugar. N-acetylglucosamine is an acylated glucosamine. You don't draw structures, but try to understand that glycosylation means addition of a sugar. Phosphorylation means addition of a phosphate. So phosphorylation happens on serines and tyrosines and threonines. There are specific amino acids that are good for the addition of specific side chains. You can have hydroxylation. We add a hydroxyl group, carboxylation. Then we have ubiquitination, which is the addition of a whole ubiquitin protein to your protein. And there's more. Okay, there's myrcylation. There's more different modifications. You don't need to remember them all. I do want you to remember phosphorylation and acetylation, methylation, because we had that with the histones. Okay, so those are important for those functions. Okay, so why modify proteins? The reason why, there's many reasons why you would want to modify a protein. One of them was turning a protein on and off, which is something we do with oncogenes. We can turn them on and off. They sometimes require a switch to be turned on and off, and if you mutate that switch, it might be permanently turned on. But when we add a sugar, it's usually to direct a protein to a specific organelle. So if you want to have your protein go to the ER or to the Golgi, then you add sugars to it, and then it gets targeted to specific area. So here you have in this case, the protein is made in the ER, but it gets different sugars added to it that then allow it to go to the Golgi apparatus. You don't need to remember what goes where, but I want you to know that these modifications help the cell decide where it's supposed to go. Okay, so if you want a protein that goes and attaches to the membrane, you may want to add something hydrophobic to it, right? Because why would a protein even associate with a membrane? If you put a lipid on it, you can anchor it in the membrane. Okay, you can't just randomly anchor a protein unless you modify it. So those modifications have really important functions, and that's why most of our proteins are modified. Which, okay, so if we make our protein in E. coli, so okay, and one example is the spike protein, okay, from the COVID virus that we talked about before. When I make this protein, and in the beginning, we tried to make this protein E. coli, just to make it as a positive control for your rapid test. Has anybody taken a rapid test? Okay, so the rapid test is like a pregnancy test. You have two lines. You have a control line and you have your disease line. Okay, and the control line, if that one doesn't line up or doesn't light up, that means your test is not working. Okay, so you always have a control. So similar to that, if we want to show our tests work, we always have a positive control. So we made the spike protein in E. coli to have it as a positive example. So can our antibody detect the spike protein? Yes, it can. Now I can detect it in your saliva. But I need to prove my assay first. So we made it in E. coli, but E. coli uses different post-translational modifications. So it doesn't work. We can't make it in E. coli and then use it as a positive control, because the spike protein that humans make, that the virus makes through human host cells, looks very different. So the spike protein in humans is super modified and has a lot of different sugars on it. So now if we want to make the spike protein as a positive test, we have to make it in humans. So between organisms, these different post-translational modifications can be very different. So sometimes when we look at a protein and want to figure out the function, we have to make it in the actual organism that it's being made initially, which is a pain. So working with human cells, E. coli, they divide every 20 minutes, human cells every 24 hours. If you want to think about how fast I can make things in E. coli, really fast. Human cells takes forever. So there are advantages of working with bacteria, but the disadvantage is always the modifications. Okay, so this is your P53, which we talked about in the cancer section. And these are your post-translational modifications here. Okay, all these up here in this line and down here, these are all modifications. So you can see how highly modified proteins are in the cell. And then you can see even more crazy is how many modifying enzymes there are. So this is your Lex layer up here. Okay, so here in the middle is your protein. Then one up and one down. Here are your modifications. And you can see there's a serine 6, which can be phosphorylated. What do we have here? Threonine 55, also phosphorylated. We have different glycosylations down here, ubiquitinations. But these are all the proteins that do that work. And then these are the proteins that interact with those specific sequences. So modifications are very important to direct any of those interactions. Okay, so here's your P53. Remember the job that it has. It kind of is supposed to check whether the genome is in order. If the genome is not in order, it's supposed to cause apoptosis, cellular suicide, or cell cycle arrest to lead to DNA repair. Now, if you think about all these modifications, if you mutate P53, it's not just a change in primary amino acid structure, it's also a change in how it can be modified. Okay, let's say you need to phosphorylate it to turn it on and off. If you mutate it into an amino acid that can't be phosphorylated, that on and off switch is gone. Okay, so sometimes the mutations have a more indirect effect downstream. Okay, we can go over this one, but I think then we're probably good. So what do proteins then do after they're being made? Proteins have many functions, and I think you've learned most of these in the BOFA section. But remember, they can have, depending on what function they have, they might need different modifications afterwards. So they can recognize signals, they can locate to many different spaces, they can be involved in anything here, in anything that's listed here. Maintain pH, provide structure, act as a messenger. So proteins then have many different functions in the cell that they have to do, and they have to be made properly from mRNA, and they have to be modified properly in order to be able to do those functions. And with that, I think we're probably okay for the day because we'll switch. This section now would take too long and I don't want to keep you too long. So with that, I'll see you on Wednesday. Thanks, everybody.